are anaerobes. Anaerobes are catalase negative. Can't break down hydrogen peroxide, no bubbles. There is a slide in your PowerPoints. I think it might be either the bacteria classification PowerPoints or the chapter 6 PowerPoints. I don't remember which one. But remember one of those PowerPoints I showed you already has a slide, and it's got the bacteria in a little puddle of hydrogen peroxide. One type is bubbling and the other type is not. The catalase test is in one of those PowerPoint slides. Okay, what do you want to remember about catalase test? All Enterobacteriaceae are catalase positive. Anaerobes are catalase negative. Streptococcus and Enterococcus, which is a streptococcus, are catalase negative. Staphylococcus and Micrococcus, which are all staphylococcus, are catalase positive. The catalase test is not done for Enterobacteriaceae. It's not going to help you differentiate one Enterobacteriaceae from another. It's really done for the cocci. Oxidase test. All your Enterobacteriaceae are oxidase negative. If it was oxidase positive, your bacteria would have turned blue. When you put this reagent on it, it would have turned blue. Okie dokie. What else was I going to tell you about? Okay, let's do this. You know, family Enterobacteriaceae are gut flora. Might be human gut flora, particularly the bowels. That's why they call them coliforms. Friendly lactose fermenters, coliforms, normal human coliforms. E. coli and his friends. Animals, their Enterobacteriaceae are included in that family. And on your list you have Escherichia, Salmonella, Shigella, Proteus, Klebsiella. You need to know which ones are Enterobacteriaceae. And here's two more I told you about. You might want to throw them on your list just so you have them there. Citrobacter is also Enterobacteriaceae and also is Acinetobacter. So these two are also Enterobacteriaceae. Enterobacteriaceae, section 5, all facultatively anaerobic gram-negative tiny bacilli. Do you think those are the only bacteria in your poop? Hell no. That's a group of bacteria in poop. But there are other bacteria in poop, like Bacteroides, probably some Staphylococcus, Lactobacillus, all the ones they said in the movie, right? But remember this one, Enterococcus. It also has entero in the name. It's not in the Enterobacteriaceae family, but it is in your bowels, and it's a coccus, gram-positive coccus. It's not a facultatively anaerobic gram-negative rod. It's a mostly anaerobic streptococcus, right? And VREF, vancomycin-resistant Enterococcus faecium, or fecalis.
So what again, Enterococcus
is the gram-positive cocci, division 2, section, what is it, 12 or something like that? It's a totally different division, totally different section, but they both occur in poop. That's why it's got entero in front of the name. Let's talk about how we're going to differentiate one Enterobacteriaceae from another. If you want to work just with this family, so let me back up a minute. So sometimes I'll say enterics. Sometimes when I say enterics, I'm referring to just this family. But sometimes when I say enteric, I'm referring to all the enterics in poop, including these guys. Okay? So this is kind of a general term, can be used either way. So when you read your question, it should not be confusing. It'll say enterics. Which of the following enterics is not gram-negative? Which of the following enterics is gram-positive? Or which one of the following does not belong to the family Enterobacteriaceae? So the question will be clear. But just keep in mind, they're all enterics, but Enterobacteriaceae is a specific family of enterics. Okay, what was I talking about? Okay, so let's talk about family Enterobacteriaceae. The friendly ones, like E. coli, ferment lactose. The ones that are not friendly, Salmonella, Shigella, Serratia, probably a few others in there, they don't ferment lactose. And why am I saying ferment? Because they're facultative anaerobes, right? So they can do fermentation reactions too. What do Enterobacteriaceae eat? They eat what we eat. They all eat glucose. They all can ferment glucose. They may digest it aerobically, they might digest it anaerobically, because they're facultative anaerobes. But they all are glycolytic. They can all use glucose. They're all glycolytic. Glycolysis. They all split it. So they can break down glucose.
Every single one.
Only some of them can break down lactose, the friendly ones. Let's talk about amino acids. Do we eat amino acids? Do we have amino acids in our digestive system? Absolutely. We eat proteins, break them down to amino acids, and guess what? The bacteria eat them too. Some Enterobacteriaceae will eat some amino acids, and other Enterobacteriaceae will eat other amino acids. So we can also differentiate them from one another on the basis of which amino acids they ferment. So in your API test strip, you have sugars and amino acids for the most part. And we're going to read the result of a color change. If we got a color change, that means the bacteria ate or fermented or whatever, used the nutrient, produced a waste product, and the waste product is what turned the cup a different color. Because we can't identify bacteria on the basis of their hair color, their skin color, their eye color, and their size and shape, really. In that strip, instead, we're looking for enzymes to run metabolic reactions. If they have an enzyme to do a metabolic reaction, that means they have a gene for that enzyme. So we're looking at their genetic characteristics, just like eye color is a genetic characteristic. Well, the enzymes that bacteria produce are a genetic characteristic we can use to identify them. I want to read you a little story.